message. Are you still alive? I'm like, I'll see you in March. Okay. So we will go over immunology, the lecture, the PowerPoint for immunology, and then I do have a review sheet on Blackboard. I don't know if you printed it off or not, but I'll go through the review sheet for test 1 when we're all done, and tell you all that good stuff too. So, immunology, this really should just be review, because you had a whole class on immunology. So you know what your immune system does, right? Your body sees a foreign invader and your body builds an antibody against it. The antibody binds to the foreign invader, and then your macrophages come along and eat it, right? Okay, so in blood bank it's very easy, because when you were in immunology you talked about the Epstein-Barr virus, and you talked about the rubella virus, and you talked about this virus and that virus. Blood bank, your antigen is always the red cell. Always the red cell, okay? Your red cells have many, many antigens on them. Some can elicit an immune response more than others. Okay, so that we do have kind of a numerical order as to who's who. Okay, as I'm going through this entire class, one thing I want you to remember, and I'll remind you over and over again, they didn't read the textbook. The antigens and antibodies didn't take the class. So sometimes they do things other than what the textbook says. And you have to always remember that, okay? That they don't always act like they should. So you've always got those outliers. So I can't stress to you enough, as a young tech, a new tech, you're not going to know about those strange things that they do. You have to ask. Our techs that are out there right now from last year's class are getting into trouble because they're not asking. It's not that they're not doing it right. They're not asking how to do it right, or they're not asking what is this doing, all right? So they're not yelling at you because you're asking. They're yelling because you're not asking. Okay, blood bank is very important. All right, so you have to do that. Okay, so what's a definition? An antigen, of course, is a foreign invader. In immunology, you talked more about those viruses and everything else. Or a naturally occurring cell marker. In blood bank, it's that naturally occurring cell marker. It's the antigen on the red cell. That's the bad guy. Okay. Again, innate immunity, you probably talked all about innate and acquired and all that good stuff in immunology. No big surprises there. Okay. Again, if the pathogen outsmarts it and your lymphocytes recognize it and mounts a response, and again, it has memory. Okay, so that's what we worry about in blood bank, is that memory. That remembering that, oh, you got a blood transfusion 2 years ago. Oh, I remember that antigen on that cell that came from that other donor, and now I'm going to wage a war against it. Okay, you guys learned in immunology, right? Sometimes it's immediate. Sometimes it takes your body a little bit of time to kind of get their sources together and then wage the war. Blood bank's no different. Okay, some are immediate and some are delayed. That's what we call them in blood bank, immediate and delayed. Your phagocytes, your cells that eat the antigen that has the marker on it. Okay, again, shouldn't be anything different. Your lymphocytes, your T cells and your B cells. So again, we worry about your B cells because your B cells are the ones that produce the antibody in your blood bank system. Okay. So your B cells, remember, those are your B lymphocytes that turn into plasma cells that produce antibodies. Hopefully this is review. Heard this before? Okay. Again, your B cells make your antibody that bind to the antigen. Okay. Oops, I'm sorry. I don't know what I did. Complement. Do you remember talking about complement? Did you talk about complement in serology, right? And the complement cascade, which is very similar to the cascade in coag. Okay. We worry about complement. Again, the end product of your complement activation is cell lysis. Well, if we gave you a unit of blood because you're anemic, is it a good thing if your body starts lysing it? No. Okay, so we need to be careful of that. We don't want to activate complement. We don't want to start that cascade so that your cells are going to rupture. All right? So, again, causing cell lysis of the pathogen. In this case, it's your red cells. In the classical pathway, that's the one we worry about with blood bank because it binds to the antigen. Okay? So, what do we do in blood bank to get rid of it? If we can stop the complement before it even becomes a problem, that's what we want to do, right? We don't want to start a reaction. We don't want to look for it. My page is stuck. Oh well, okay. So, storage. Complement deteriorates if we store it at refrigerated temperature. So if we stick it in the refrigerator, complement will deteriorate. It won't become a problem. Okay, it won't cause a reaction in our specimen. Anticoagulants are anti-complementary, which means if I use a Vacutainer tube that has an anticoagulant in it, it will automatically inactivate complement. Will not be a problem. What color top tube did I give you in class on Tuesday? Do you remember what tube I gave you in class on Tuesday? Lavender. What is the anticoagulant in the lavender top tube? EDTA. So EDTA is anti-complementary, which means it causes the complement not to get activated. So in blood bank, we want to use an EDTA tube. Okay? Now, we can use the purple top tubes. There's also an EDTA that is pink. I don't know if you've seen that in your lab at your job. Okay, they're usually bigger. So a lot of labs will use the bigger pink EDTA tubes for their blood bank. You could still run a CBC on it. It isn't any different. You could still run a CBC. But again, they're usually a little bit bigger, plus then they're marked pink for blood bank. Not all the labs do it. Our lab doesn't. Yes, ma'am.
Did you need that more blood? Do you need more blood in blood bank than you would for CBC or anything?
Yes, yes. Not for a routine. If your patient is perfectly normal and everything goes as planned, you can do it from a little purple top tube. Okay? But what if they have antibodies? What if there's a reaction? And then, of course, you need to do more testing, so you need to do more. Okay? Now, in 98% of the time in blood bank, everything will go smooth. It's that 2% that are going to pop up at 2 o'clock in the morning when you're in the lab all by yourself, okay, that you're going to have your problem. And then worse come to worse, you have them go draw you a new specimen. Because again, should we draw, 10 tubes for that 2% that may need it? No, we're not going to. Okay. So unfortunately, I'm going to spend 95% of my time on that 2% of the problems. But then you'll be good at it, right? Didn't Diane always try to tell you, at least if you knew what was normal in your red white cells, then if something abnormal would pop out at you, you'd go, whoa, wait a minute, that's not right. So again, that's our goal. And again, we can heat it if we heat the serum. So if we drew a clot tube, which did not have an anticoagulant, which did not inactivate the complement, we could heat the serum for 30 minutes. If we accidentally stuck our serum in the incubator and we're like, oh, shoot, no, we wanted to find that complement, do you know how you would reactivate it? If you killed it with heat, do you know how to reactivate it? I'm sorry?
Cool it off?
Well, you would have to cool it back down, correct, but that wouldn't reactivate it by itself. All you have to do is add a very small amount of fresh serum, and it was likely to reactivate it. So, rule of thumb, never heat the entire sample, right? Always aliquot and heat a small sample, okay? Because you may have to, for some reason, reactivate it. HLA antigens. As an MLT, you are not responsible for the HLA, all right? The only one that I know that anybody's ever even asked a question about was the chromosome 6, which is the one that is associated with blood bank. But as an MLT, you are not required to know anything about the HLA, so I don't go into great detail about it. But for those of you that are going to sit for the registry to maybe perhaps get your MT, HLA is something that you're going to want to have to review as far as where it's going for that. Platelet antigens. Platelets do have antigens just like red cells and white cells. Okay, so they can cause a problem. On your take-home test right now, there's a question about NAIT, about the neonate alloimmune thrombocytopenia. So again, when your immune system attacks your platelets, so if your immune system attacks your platelets and the macrophages come along and eat your platelets, what's going to happen to you? Exactly. Petechiae, and you started having a problem with bleeding. So with newborns, do we want that? Okay, some characteristics of some antibodies. I really wish I knew why my papers got all messed up. Oh well. So they are immunoglobulins, right? Immunoglobulin antibodies, exact same term for the exact same thing. And they are in response to a stimulus of a specific antigen. They can combine with that antigen. You learned in immunology that they're lock and key, right? One antigen for one antibody. That's kind of true in blood bank. Sometimes they're so close they do tend to get a little confused. So we can get discrepancies because of those that are a little confused. Again, they didn't read the textbook, and sometimes they just forget what they were supposed to do, and they just want to wreak havoc with you. So that's when your lecture on Monday will be discrepancies in the ABO blood group system when they're doing what they shouldn't do. So again, polypeptide chains, IgG, M, A, D, and E. No big surprises, right? Gamed, right? So that's their molecular structure. They have heavy chains and light chains and all that good stuff. And again, just review. We don't really worry about all this in blood bank. We just worry about antigens and antibodies, not so much about their structure. In blood bank, we're going to talk about IgGs and IgMs. IgAs will sometimes play a role, but that will be like in transfusion reactions, which is several weeks away. So we're going to worry about IgGs and IgMs. We don't worry about Ds and Es, okay? On your test, you're going to have a list of characteristics, and you're going to tell me, is it IgG, IgM, both, or none? Okay, so as I'm going through this, kind of think about that in your head. So here's a nice comparison scale of your IgGs and your IgMs. So again, the IgG is a monomer, means it's just that one little Y squared antibody. The IgM is a pentamer, much bigger, right? Okay. Dithylthetriol is a reagent that we can use as an aid to separate out IgM from IgG. If I've got agglutination in my tube, can you physically see the antibody? No. We learned that in immunology, right? You had to add something to the reaction so you could physically see it. So we can't see them. So if we have a reaction, we have agglutination, and we don't know, is it IgM or IgG? I can add dithylthetriol, it's just a reagent, and it will bust apart my IgM. So if I bust apart my IgM, what's going to happen to my agglutination? It's going to fall apart. Correct, right? So you would go from having agglutination to no agglutination, where your IgG, is it going to affect your IgG? No, so you're still going to have agglutination. Make sense? Okay. Does the antibody bind complement? Both of them do. When we get into other blood group systems, certain ones will bind more than others. So that's something you're going to tell us then. What crosses the placenta? Talk about this in immunology and the importance of what it means, right? So if I have a mom, and you have to remember, in blood bank, really what we're worried about are, well, more than this, but I mean, one of the things we do worry about are moms and babies, right? Moms and babies. You have to realize when you're pregnant, that baby is only half you, right? So you are carrying a very big antigen, right? And so you can react against that baby. So if we draw mom's blood and we do an antibody screen, which means it's positive, she has an antibody in her plasma, We need to know, is it IgG, because is it going to cross over to the placenta and attack the baby? Is it IgM? Are we going to worry about it? No. Okay, so again, that's why we need to know, is it IgG or IgM? What is the identity of this antibody? Again, some antibodies are more specific than others as far as attacking, hanging on, and causing a problem. And we're going to learn about those in numerical order. ABO is number 1. RH is number 2. When you tell somebody your blood type, what do you tell them? You tell them I am B positive, right? B, ABO blood group system, most important. Positive, RH system, second most important. Do you know whether or not you're Kel positive or not? That's third, and you're going to know that before you leave my class. Okay? But again, we don't tell people that. Oh, and I'm Kel negative. Right? Okay, but you will. Stimulation. How are the antibodies stimulated? Again, in blood bank. IgG are stimulated either by a blood transfusion or by pregnancy. Do men get pregnant? So if a man has an antibody, what type do you think it is? Chances are it's IgM, right? Unless if they've not had a blood transfusion. So IgM in the blood bank system are naturally occurring, which means something else has triggered it. Gut bacteria is a big one. Okay, some other antigen, because they're just a little confused and they've made an antibody that will attach to a red cell, but that's not really where it came from. Okay, naturally occurring. Reactive temperature. IgGs like heat. 37. You noticed in the room I have incubator baths, or not incubator baths, little incubators. There's 2 on each side of the room, okay? Starting next week, we're going to need to make sure we turn those on as soon as we get to the lab, or even if somebody's in there first thing in the morning, okay? We need to turn those on because we are going to start doing 37. So we have to do our reactions at different temperature phases. If I do a reaction at room temperature, what temperature is room temperature? 20 to 25, right? So if I'm doing a test at room temperature and I'm getting agglutination, what type is it? IgG or IgM? IgM, right? We did reactions in class on Tuesday. Were we sitting in an incubator? Did I have any incubators out? Did I make you walk over to the incubator? We did them at room temperature. So what antibodies in your ABO blood group system? IgM, they're room temperature reacting, okay? RH antibodies, remember we did the D in the tube? Those are your RH antibodies. We did those at room temperature as well, right? They're actually IgG. So you're like, well, wait a minute. You just told us IgMs are room temperature, but you told us we could do that and it's IgG. When I asked you what was in those bottles, when I said what was in the blue bottle, what did you tell me? Anti-A. Yellow bottle was anti-B. And I said one was A antibody, the other one was B. And I said, okay, what is in your anti-D? And you all said D antibody, right? And I said and you all did exactly that. You all stared at me. And I said potentiator. And at first you're like, what the heck does that mean? And I said, never mind, we'll get there, right? A potentiator is something that will cause a reaction to happen.
So it's a catalyst?
You could think of it that way. Yes. It's going to enhance the reaction. So if RH is IgG and really should only react at 37, but I want to be able to do it quickly at my table, I have to give it a little extra juice, right, to get it to react at my table. All right. In the RH blood group system, which we'll talk about next week, just giving you a little forethought, okay, some anti-D will react at room temperature with a potentiator, and some will not. Okay, so even though today you might say, oh, it's negative, It may truly not be negative. Okay, so again, we're going to go over that. They're called weak D, so we'll go over that next week. So our reactive temperatures are going to mean something to us. If I have a reaction and it's coming up good 3+ at room temperature, I'm pretty sure it's an IgM. Okay, if I have to go stick it in the incubator and do a little extra special work, it's probably an IgG. Okay. Reactive phase, AHG or saline. AHG is anti-human globulin. It's and again, this is ahead of schedule too, because we'll go over your reagents in a future lecture. Your IgMs are pentamers. They're big, right? They have 5 antibodies sticking out there. Your IgGs are just that one little one, right? So if I can take antibody and hook it to that IgG to make it bigger, like your IgM, it's going to react better, right? And that's exactly what the anti-human globulin is. It's an antibody against an antibody to make that structure larger. Okay? Saline means it doesn't have anything in it other than saline. So it's not going to change the antibody. Do I need to? IgMs are already really big, right? I don't need to give it anything else. It can already reach. Okay? And then the response, this is right back to your immunology. Your IgMs were first, right? When your body saw the foreign invader, it produced IgMs. Those were your primary responders. IgG were your secondary, right? Which did you have more of, IgM or IgG? Do you remember? I'm sorry? And no, after your reaction happens, you've got your IgM first and then your IgG. Which did you end up having more of? IgG. Exactly right. Exactly right. So again, I'm going to have the characteristics listed on your test. You're just going to tell me, is it IgG, IgM, or both, or none? Okay. Types of antibodies. An alloantibody. An alloantibody is an antibody that you're getting because you've been exposed to an antigen. In blood bank, it's either through pregnancy or it's through a blood transfusion. Okay, it's an allo. It's from someone else in the species. Pregnancy or a blood transfusion. Autoantibody, what's an auto? Against yourself. Exactly right. So your body has built an antibody against your own red blood cell antigen. And we can detect that. Okay? Naturally occurring, again, often IgM develops without any known antigenic stimulus. We don't know. Sometimes, a lot of times it's gut bacteria that are just very similar. Okay? So again, untransfused male with an antibody, what type is it? Okay. Blood group antigens. Okay, in the ABO blood group system. And we're going to go about this in great detail next week. You'll have an entire lecture over ABO. This is just kind of a thing. In your ABO blood group system, so think of your red blood cell as a balloon, okay, a nice smooth surface, all right? But then attach spines to it, okay? In your ABO blood group system, you have your red blood cell, and then you have your antigens that are protruding from the red cell. The antigen is sticking out there. So if it's just me and I have no arms and you walk by within 2 or 3 inches, am I going to be able to touch you? No, right? But if my hands are stretched out and you come within 2 to 3 inches of me, will I be able to touch you? Absolutely. So if your antigens your antibodies are floating by in your plasma, and I have antigens sticking out there, does it not make sense that the antibody can find the antigen easier? It is, right? So in your ABO blood group system, if my blood is different than the antibody you gave me, my blood can reach out, grab it, and boom, we have a reaction in the ABO blood group system. Okay, it's easier. It's one of the most important ones because it will do it. All right, and they protrude from the red cells. The Rh antigen are actually embedded in the membrane of the red blood cell. So again, if it's embedded in the membrane and that antibody floats by within a couple inches, is it going to be able to grab it? No, it's within the membrane. So we have to do something to get it to glob on, right? Potentiators, right? Okay. There are soluble blood group substances. Some of us are secretors and some of us are not. All right, this is a whole nother lecture too. But we can actually find out your blood type from your spit or your saliva. So that's kind of CSI, right? You got that little guy that's out there and he just committed a murder, but we can't really prove it to him, so we stand outside in his workstation and lo and behold, he goes and takes a smoke break and spits, and we can go, after he walks in, gather his spit, and voila, we can find out his blood type, right? Not only his DNA and everything else. So some of us are and some of us aren't.
I thought you would have blood
For what?
to do
Nope, we can find it in your saliva. We can find your antigens in your saliva. If you are a secretor. Whole nother lecture. So when we talk about other blood group systems, if that fascinates you, then pick Lewis. Okay? So if so we talked about the ABO antigens are actually protruding from the red cell. Rh antigens are part of your red cell membrane. With the Lewis antigens, because they're in your secretions, okay, they are adsorbed onto the red cell. So in blood bank, we have absorb, AB, and adsorb, which is AD. The only way I can explain it, and I don't know if it'll help you or not, if you have a puddle on your counter, you grab a sponge, what does the sponge do to that liquid on your counter? Absorbs it into the sponge, right? If I have a nail on this table, and I take a magnet and put it over that nail, the magnet picks up the nail, right? Is it absorbed into the magnet or is it just adsorbed to the surface? That's the best way I can explain the difference between absorb, which actually goes into the sponge, and adsorb, which just kind of sticks to the outside. So again, we're going to talk about absorptions and adsorptions through the thing. Okay. Antigen production occurs during red blood cell maturation in the bone marrow. Not all antigens are well developed at birth. You're going to tell me this not next week, but the week after when we talk about other blood group systems. So those are some of the ones that you're going to be asked to do. LE is Lewis, P, and what happens to the people with P? You're going to know that one forever, aren't you? Proximal hemoglobinuria, yes. LU is Lutheran, which is Lutheran A or B. SDA, MNS, the well-developed antigens, so well-developed when you're born, RH antigens. Again, this is why moms have problems when they're pregnant. And if you're RH negative, you have to get that RhoGAM shot, right? That's your take-home test. Then Cal antigen, Cal is third after ABO is number 1, RH is number 2, Cal is number 3. Then FYI, FYA, so it's again FYA and B. Duffy is FYA. I don't know where the Duffy came out of that, but he did. Okay, Duffy, and Duffy is number 4. Then you have JKA, and that is kid. Okay, kids are special antibodies. Again, the antigen is well developed at birth, but the antibodies are special in that, just like little kids, they like to run and hide. So kids will cause a problem for you. Gee, does that sound familiar, Jen? So when we talk about other blood group systems, maybe you want kid, huh? Or maybe you don't. Run as far as you can. But kids like to run and hide. They are a problem for us in blood bank. So, and again, that's the best way I can describe it to kind of help remember. blood group antibodies, again, blood plasma produced after a foreign antigen, immunoglobulin class, IgG, IgM, some IgA, but again, that's transfusion reactions, which is a while away. Subgroups of IgG exist and IgA exist. We're not going to do anything with the subgroups, but again, I want you to realize they're out there. And if the antibody isn't doing what it should, it could be that it's a subgroup. And at a lot of points, you just have to send it to a reference lab because you don't have the reagents to figure it out. Okay? There's only just so far we can go. And even less and less are in blood banks now, more going to reference.
When you have issues where it's an emergency and you start having issues with that, you just give them O negative to be on the safe side because you're not sure what they're having a reaction.
No. The only time you can give O-neg without a compatible cross-match, okay, is in emergency release when the doctor signs away his life and says, I will take everything, I'll be responsible, okay? Or if you cannot find compatible blood. Then we go least incompatible.
Wouldn't that be considered incompatible?
Possibly.
Because you're unable.
Or you send it to reference and hopefully they can find somebody that'll get blood for you. Again, it's going to be one that causes a problem.
But if it was in an emergency situation that starts bleeding out, and you got one of these where it's a subclass reaction, that you couldn't tell what the class was, so that you couldn't find it.
Again, if the doctor wants emergency release,
Okay, so what would be your call? That would be the doctor's.
you betcha. There's very little that's your call. Yeah. Yep, they make the big bucks for that. So many times we walked up to a doctor who is demanding emergency release blood, and as soon as you take that form to him that says you have to sign this and say that you're accepting responsibility, all of a sudden, okay, I can wait for the cross-match. It is amazing how that works. Okay. So some factors that determine antibody production. Again, these are some things you should have talked about in immunology. So antigenicity or immunogenicity. The amount of antigen on the red cell. If it's just one antigen, what's the likelihood of your immune system recognizing it? But is there just one antigen on your red cell? No, you have lots. Presence of similar antigenic substances. A lot of times, like I said, gut bacteria. The number of red blood cells introduced. So again, did you just cut your finger and have your neighbor cut their finger or your little friend and you just did blood brothers? Or did you get a blood transfusion where you got 450 mils of red blood cells? How much was exposed to you? When you're pregnant, you have that placenta. So blood never really crosses between mom and baby, right? Not the red cells. Kind of like your kidneys, right? It'll flush out everything else, but it'll keep those red cells in. But what if there's a small tear in the placenta? Right? You have that baby, that placenta is attached to the inside of the uterine wall. Once it comes off, you have a very large sore that's open, right? And what have we learned about bloodborne pathogens? That's when they get in when there's a break in the skin, right? So that's why the first baby usually isn't a problem, because it didn't cross over in the placenta. But then as soon as that baby's born, then baby's blood can then go into that nice hole that's in mom's uterus. Second baby's like, aha, we've seen that before. Let's wage a war. Again, that's your take-home test. A large infusion may paralyze the immune system. So you were talking about emergency release, right? If we have a patient who needs a massive blood transfusion, a definition of a massive blood transfusion is where you, if your body, let's say, technically holds 10 units of blood, which means within 24 hours you've had 10 units or more, is a massive, what we consider a massive. So in other words, we've exchanged your entire blood because of you're bleeding out and we're giving you more. Okay. At that point, your immune system's like, I don't know, you're giving me too much, I don't know what to do. All right, so again, that's what the saving grace within a mass transfusion is. We're giving you so much so fast, your immune system isn't going to react, so we don't have to worry about those incompatibles because your immune system just doesn't know what to do.
How long is that?
Good question, not exactly sure.
It's not a permanent situation, correct? Like they're not going to immunodeficiency.
Well, you're not going to keep transfusing that much blood, that person's either going to die or you're going to run out of blood, one of the two. And now, when 10 years down the road and you do a blood transfusion, and get less blood, then his body may have a reaction then. But yeah. Route of introduction, again, blood transfusion, big unit of blood, 450 mils of blood. Maternal fetal bleed, well, now you're just talking about a little tear in the placenta. Number of exposures, how many times have you been pregnant? Because it's an exposure every single time, right? Even though it's the same dad, are they going to inherit the exact same antigens every time? No, you're only getting half of his DNA, right? You're only getting half. So your babies aren't exactly the same. I don't know how it works with twins, but that'd be interesting to know, to see what antigens they inherited, to see if...
Mine are identical, so I'm going to assume that
it would be the same.
because they.
Although
because the
identical...
eggs that- But they aren't. Obviously, they're different people, so I don't.
Right. And I know that identical twins are a lot of times a good match for an organ donation, but they're not always an exact match. Which it seems so odd that somebody else out there could be a better match than you would think of your identical twin. But that is interesting. And I've read that too. So, okay. Immunological state of the patient. Again, newborns do not have antibody. Okay, to the ABO blood group system. They don't have antibody. And then your immunodeficient state. So if you have a person who has an immunodeficiency problem, and what disease do we know of that is? Acquired immunodeficiency syndrome. AIDS, right? HIV. And there are other people that have immunodeficiencies. That's not the only disease out there. Antigen-antibody reactions in vitro. In vivo is inside your body. In vitro is in a test tube, right? So we detect it by visible agglutination or hemolysis. Both are positive reactions in blood bank. Clumping, like what we saw on Tuesday, or when your red blood cells rupture, which is the artificial way I had you when I had you do water. Remember, in your red cells and your plasma all turned to Kool-Aid, right? Hemolysis. Those are your two positive reactions in blood bank. What's going to cause the hemolysis? And what's going to cause that? Complement. Yes, yes. Okay. And agglutination, again, antigen-antibody reaction. Hemagglutination occurs in 2 steps. First step is sensitization. Second step is the lattice formation, or the actual agglutination itself. So sensitization. That's basically where the antibody binds at just one site. So think about those monkey bars, okay, and you're just hanging on with one hand, okay, binding to one site. The antigens and antibodies will combine, but at this point they are reversible. Because they're only hanging on with one hand, they'll easily come off. So it's just sensitized. Remember I told you you're not going to kill the person, you're just going to sensitize them? Okay, so they can come off. So factors that will affect sensitization. You need the correct serum to cell ratio. What cell suspension did I have you making on Tuesday? What was that big lab that we did on Tuesday? 2 to 5%. So we don't want it too light and we don't want it too heavy. So what am I grading you on in lab today? A visual 2 to 5%, right? That's very important. Okay, very important that you can do that because it affects it, the sensitization. Serum or plasma. So in the serum, in the testing that we did in the lab, we used reagent sera, right? The anti-A, the anti-B, the anti-D, that was reagent. Or we can use the patient's serum or plasma, okay? As a rule of thumb, and please ingrain this in your brain, because you're going to ask me 50 million times, and I will answer you every time because it is important, but try not to. 2 drops of serum, plasma, always. 2 drops. 1 drop of cell suspension. Whether it's reagent cell suspension, patient cell suspension, it's always 1 drop of cells to 2 drops of plasma. Again, be it patient plasma, be it reagent, sera, or patient serum. Okay? In my student lab. Now, when you get out into the real world, they may reduce that. 1 drop and 1 drop. But you have to realize, some of you were laughing at my expiration dates on my reagents, right? The only way I can guarantee that we're going to get a reaction, you got to add 2. Okay? You got to add 2. Yes, ma'am?
You said 2 drops of plasma serum to 1 drop of blood.
Red cells. Cell suspension. 2 to 5% cell suspension. Whether that is reagent cell suspension or whether it's patient cell suspension. It's always 1 drop of cells to 2 drops of patient serum or serum. Okay? All right. Other factors. Temperature. Again, 37. I have 37 reaction baths in there. Incubation time. How long are you going to put it in there? And please don't jip on this. A lot of students get in a hurry and they're thinking, oh, it's only 15 minutes. It'll do me. In the beginning, I'm going to make you strong strong antibody concentrations. I'm going to get weaker and weaker as it goes by. So even though your 15 minutes might get you in the beginning, it might come back to bite you at the end. Okay, so don't jip on it. Follow your procedures very exact. Okay. And again, does depend on the test procedure. So some of them will tell you one thing with the others. pH, the best range is 5.5 to 5.5 to 8.5. pH is all regulated in your reagents. That's not something we have to do. The only time we would change the pH is if we had an antibody that wasn't doing what it should be doing. All right, IgM is the main one here. You would want to regulate, make sure your pH is very close to 6.5. That's where its optimal pH is. But again, you're not going to do that, and you don't have time for all that. Ionic strength of your reaction medium, okay? We use something called LIS, low ionic strength solution, LIS, okay? And it makes a difference is what you use. So again, but you will have no visible agglutination. You centrifuge this tube, you will not have any visual agglutination. So that's sensitization. Again, your antibody is binding to your red cell in just one site. So your lattice formation. That's where your antibodies cross-link, okay, to your cells that are coated with antibody, causing visible clumping. Your cells are causing agglutination. Factors that affect your lattice formation, the immunoglobulin class. We call IgGs incomplete because they're just little guys and they can't form it themselves. Okay, remember I said AHG, right, which we'll talk about in just a minute. IgM are what we call complete antibodies. Another thing, if I were taking the test on Monday, next to IgG, I would write incomplete, I would write warm. You've had it in other PowerPoints, but this will just be a place that you can put it again to remind you. IgGs, remember, need warm. IgM are antibodies we call complete, which means they can complete the agglutination between 2 red cells because they're big enough. So I would also write on here, if I were taking the test on Monday, complete and cold. It's not that you have to stick them in the refrigerator, although the refrigerator will enhance the agglutination. When we say cold, we're talking room temperature versus warm, which is 37. Okay? 4 degrees will really make them clump. The number of antigenic sites. If I've got more antigens out there to grab, I'm going to grab them and I'm going to have an agglutination reaction. Location of the antigen sites. Remember, your ABOs, where are they? They're protruding from the red cell. They're easy to grab. RH, where are they? In the cell membrane, right? They can't go by, they can't grab them. Okay, so again, makes a difference. And then zeta potential. And we'll talk about zeta potential in just a minute. So again, agglutination with an IgM can bind a bunch of red cells all by themselves. IgG, not so much. Possible problems with your antigen-antibody ratio. Did you talk about prozone in immunology? Okay. So again, when your antibody is present in excess. So sometimes in blood bank, and this is way ahead, we'll talk about this more in the future, but you can have a lot of antibody that's not reacting very strongly, so you can dilute it out, and even though your dilutions, you'll still get 1+, 1+, 1+, because you just got so much, but that's weeks away. So again, what are we going to do? We have to use our 2 to 5% cell suspension, okay? And if we suspect a problem, we can titer our patient. But again, that's weeks away. It's actually in your take-home test. So techniques to enhance agglutination. Some things we are going to do in blood bank to make those reactions happen, or at least to rule them out because they don't happen. Centrifugation. Obviously, if I'm centrifuging that specimen, it's forcing the red cells together, causing the red cells to match up with that antibody, and then they're getting the agglutination. Did we not put it in the centrifuge, and did we not spin it for 30 seconds to get our agglutination? Exactly right. Albumin. Albumin is a positively and negatively charged molecule that will help with zeta potential, which we'll talk about in just a minute. Low ionic strength solution, LIS, has a sodium chloride and a lower than normal saline. You have in that box of reagents that I gave you, you have LIS. All right, you have the low ionic strength solution. In almost all procedures we do, you're going to have to add LIS before you incubate. All right, so you need to watch your procedures and know when you add it and when you don't. Because when you take it out of the incubator and you centrifuge and you're not getting a reaction when everybody else is, you're going to go, oh, I forgot to add my LIS. So what do you get to do? Add your LIS and go back into the incubator for 30 minutes. Everybody else gets to leave and you're still working on your stuff. Okay, so again, it makes a difference, so don't forget. Enzymes. I don't have any enzymes to work with you, but when you get into the real lab, they'll do some with enzymes. When we talk about other blood group systems, you're going to tell me which blood group systems enzymes enhance and which ones they destroy. So if you have 2 different antibodies or 2 different antigens causing problem, okay, if I add an enzyme and my reaction gets stronger, voila, I can look at my list of ones that get stronger with enzymes. If my reaction goes away, voila, I can look at my list of ones where the antibody is destroyed. Make sense? Okay, so they're just a helper. AHG, anti-human globulin, also called Coombs serum, is that red blood cell with antibody already attached to help bind other red cells. Crossing that link, if you will. Principles of antibody potentiators. Enhancement media. Remember I told you an anti-D, it's anti-D plus potentiator. So they promote agglutination. Okay? Direct agglutination, again, reduce zeta potential. Get those red cells together. LIS, low ionic strength solution, can be called enhance, can call immuadd. There are several answers, but if you read on the bottle, it says it's low ionic strength solution. There's saline. Serum albumin, good for RH. PEG. PEG is one that I have. PEG alters the procedure. With PEG, you only do it in the AHG phase. So you pretty much have to know what your antibody is when you get there because you don't do it through all phases. But if you kind of know what your antibody is and you're just verifying it, PEG will kind of shorten your procedure for you, all right? Because I use manufactured antibodies to give you, to find out what, for your unknowns, PEG causes your tubes to turn to jello. So they're not very good. So I don't use PEG in the lab. But so many students come back from their clinical and go, that PEG is the coolest stuff ever. because you don't have to go through all the phases. You only have to do the 37, and it does shorten your time. So again, though I have it, so there are times, I have a friend that works at the Missouri, Mississippi Valley blood donation, whatever. And so every once in a while she'll drop off some real patients for me. And so then we can use PEG because it will work for real patients, but it just doesn't work with my manufactured ones. I want you to see them. I want you to get reactions. So at first I'm going to try to give you a 3 to 4 plus. And then as the time goes on, I never try to give you less than 2 plus. Every once in a while, though, if it's heterozygous versus homozygous, I'll get some 2 plus and some 1 plus in the exact same tube. Not much I can do about that because that's just whether they're showing dosage or not. Again, enzymes, those are your enzymes, and again, they enhance RH, KID, and Lewis, but they'll destroy MNS, Duffy, and Duffy A and B. So again, you're going to tell me all about that in that lecture. So zeta potential. What is zeta potential? Okay. The best way for me to describe it is, let's say we had 2 Earths, okay? Earth is surrounded by an atmosphere, right? So if you had 2 Earths with their atmosphere, could they touch ground to ground? No, right? Kind of like a hamster and the hamster ball. Can the 2 hamsters touch each other in the hamster ball? No, the balls will touch each other, but the hamsters can't, right? But if we want the hamsters to be able to touch each other, we got to figure out how to get those hamsters closer together, right? Okay, so we need to force the 2 red blood cells. They're naturally going to repel each other because of their positive and negative charges. We have to get them closer together to form that antigen-antibody bond, okay? So you have what's called a slipping plane, which is that area in between the outer limits of the ions that move the cell. So again, we have to reduce the zeta potential. We have to break that boundary to get those red cells closer together. And that's what zeta potential is. So again, the bouncing of the 2 Earths because of the atmosphere. We want to get the Earths together and get the atmosphere out of the way. That's your zeta potential. So your red cells have a natural atmosphere of positive and negatively charged that keeps them from coming close together. Have you ever had a magnet, and when you do 2 magnets, you put them one way and they repel each other, but then you turn one and do the other way, and then all of a sudden they attract? That's what we're trying to do, is get them to attract, because they naturally want to bounce away from each other. Okay? So again, how do we do that? We add an additive potentiator, right? We can centrifuge them, right? If it's positive and negatively charged ions keeping them apart, if we added LIS, which is a low ionic strength solution, it's going to change that positive and negative charge so that they can come closer together. So reducing that zeta potential. Okay? That's it. And I should have given you guys a break, because I have to give you guys a break every 10 minutes for every hour of lecture. But in all honesty, we're done. So all I have left is review. So do you guys want to take a break and go potty and do whatever, and then we'll come back and review? Okay, go. 10 minutes. I got an email. I have to now. I'm required to give you a 10-minute break.
What is that study guide under? Is this test review 1?
Yeah, test review 1.
I'm so just over you since then.
And I would be too. And I would be too. And I would be too. So sorry. Okay, so if I were taking the test on Monday, I would know the definition of dominance recessive, what a chromosome is, mitosis, meiosis, genotype. Genotype is what in blood bank? The genetic makeup based on your inheritance, right? Mm-hmm. So how do you find the genotype?
Genotype is
Family history.
Family history, exactly right. That pedigree chart, if you will, right? Phenotype. Mm-hmm. Phenotype in blood bank is what we can actually test for in the tube, right? Okay, so we can tell them, are you A, are you B, are you O? If I can tell you you're AB, is that your genotype as well as your phenotype?
yeah.
Yes, because remember you have 2 genes, right? So if I can tell you you're AB, then that is both your genotype and your phenotype. If I tell you you are O, is that your genotype or your phenotype?
Phenotype?
Type O.
Oh,
To be O, what two genes did you have to get? O and O. So again, O is your genotype as well as your phenotype, right? An amorph in blood bank, which blood group system in the ABO is an amorph? Amorph means no detectable product. Oh, yes, yes, because there's not an A and there's not a B. So to be an O just means we did not have a reaction, right? So again, an amorph, all right? Gene is just the product that you inherit. And an allele. What is an allele in blood bank? If I inherit the KEL gene, it's the complementary, right, on the other side of the DNA. So and remember, you can get the gene from mom and the allele from dad, or you can get the gene from dad and the allele from mom. It doesn't matter. It's just if you think of those train tracks or those monkey bars, one's on one side, one's it's just its complement on the other side, right? When we talk about other blood group systems, we'll talk about the fact that they aren't alleles to each other, but that's ways away. So anyway. Review history dates and their significance. So again, if I was taking the test of all those history dates Barb talked about, which ones would I know? I would know that in 1900, Carl Landsteiner discovered what?
where is this? Do we have that? No.
You should have. Yours look like this. No, I don't have yours out. Yours didn't look as fussy as mine. Just a list of the dates. But you can look at it. It's on Blackboard too. So I would know 1900. That's when Carl Landsteiner identified the ABO blood group system. So I would know that if he discovered the ABO blood group system and kind of said, hey, you know what? Those of you that are interested in transfusions, you might want to use this to cross-match to find out whether or not your transfusion is going to work. And that happened within 7 to 10 years. Okay? So again, went undiscovered for about 7 to 10 years. They don't know exactly when. All right. I would know that in 1940, they discovered the RH blood group system. I think on your paper it's 39 to 40. I would know in 1947 was the formation of AABB.
AA and AV.
AABB, American Association of Blood Banks, was formed in 1947. In 1950, we started using plastic bags. Why did I tell you plastic bags were important? They didn't break. They also breathe. Exactly right, which helped with the pH and the life of the red cell. Magic bags. Breathe but don't leak, right? I would know that in 1967, they discovered RhoGAM, or RhoGAM was actually clinically produced, right? We could use that against hemolytic disease in the newborn. In 1970, it was an important year for blood donations. What did we refuse and what did we allow? What change did they make in 1970? All volunteer. We were no longer allowed to pay people for their blood. Why did we have to make that change?
People who are going to get paid aren't necessarily the cleanest.
Exactly. They found that donated blood, people who were willing to donate their blood, it was a healthier blood supply. Exactly right.
So you say you refuse? What? Loud.
Do what? I'm sorry?
I'm confused now. You said you was not allowed to pay people.
As a blood bank, we were not allowed to pay people to come in and donate their blood. Well, not to donate it. We couldn't buy their blood anymore. We could only take blood from people who are willing to give it away for free. We're only willing to donate it. Because people who are willing to give it away for free were a healthier population than those that wanted to sell their blood. Does that make sense? And then the last one is 1983. What was significant in 1983? That's where we could finally store it for 42 days. And that's as far as we've gotten so far. We are still at 42 days expiration. 1983. We've had it for that long. Okay, so those are the dates that I would know. How are blood group systems inherited? Are they sex-linked? Are they recessive? Are they dominant? They are codominant, exactly right. So if you get one from mom and one from dad, neither is going to overshadow the other. Okay, what is zeta potential and how do you reduce it? Zeta potential, remember I told you, was that force that keeps the red cells apart. So how do you reduce zeta potential? What were some things you can do in the laboratory to get those red cells together? Centrifugation. Adding Lys. Exactly right. Exactly right. Okay. How are antigen antibody reactions demonstrated in blood bank? Two ways. Now, how are they demonstrated? How are you going to see the reaction in blood bank? Agglutination. Right? I showed you both on Tuesday. Agglutination and hemolysis. Hemolysis. And we did it fake because we just added water and I showed you what the hemolysis reaction would be. But again, it'd be complement that would cause it. So agglutination and hemolysis. Which do you think is going to be more frequent? Which do you think you're going to see more than anything else? Agglutination. Why? Don't we try to use an anticoagulant so that the complement doesn't get in our way? So if we're going to eliminate complement by using anticoagulants, then we're not going to hardly ever see a hemolysis reaction, right? It's
Can it be your own air, which is most likely going to cause hemolysis?
It could possibly be, yes. But again, it is a possible reaction, yes. So if hemolysis is a possible reaction, do I want to use a hemolyzed specimen for my blood bank testing? No. So if you're testing in blood bank and you're going to do a cross match or even an ABO, and the phlebotomist brings you a specimen and it's hemolyzed, you're not going to use it, right? Because how am I going to know it wasn't from the original specimen versus a reaction that took place in the test tube? Not complement. So you would reject a hemolyzed specimen, and again, because hemolysis is a positive reaction.
You could lyse your red cells to do testing for other blood groups if the antigens are present inside of them.
We do, yes. That's called an elution, but that's like week 7. Yep, we do. We have to, right? We have to. Yes, ma'am.
Is book able to save my protein?
In an electronic cross match, remember we talked about an electronic cross match. This is what we've gotten to now, where if I take the blood type of the patient and I put it in the computer, and I take the blood type of the unit from the Red Cross and I put it in the computer says, oh, blood type of the patient and blood type of the unit match, you can give this blood, where the two bloods have never touched each other, okay? In the olden days, we had to mix blood from person A with donor A and make sure they weren't going to react. Now we let the computer do it. In order to validate that system, and again, you're weeks away, okay, you have to have a second blood type. It's required. That's the only time we allow finger stick blood.
So, but
For
what
A.
in the case of a baby, newborn, where they're getting on glow sticks and things like that? Or you need to do typing and everything.
That's a whole nother bag of worms.
Okay, so. it's
That's your
not-
take-home test, week 7. Okay, so you're way ahead. Okay, but again, a finger stick blood would do you no good other than an ABO, and that's the only time we use it is to confirm the ABO from a previous time. Okay, but you're way ahead, which is good thinking. But anyway, where are we? Antigen antibody reaction, hemolysis, and agglutination. Agglutination more frequent. Calculate gene frequencies using the Hardy-Weinberg equation. So that equation that we did yesterday and that extra sheet that I gave you that I still have more of if somebody wants to play. But that's a question. And I think that's worth 5 points.
What was that equation? How did you phrase that?
The Hardy-Weinberg equation?
Yeah. It was something times something plus the first and the second.
It's the gene frequency. You're talking about the formula?
Yeah.
What are you talking? The gene frequency squared plus 2 times the gene frequency and its allele plus the allele squared, right? P squared plus 2PQ plus Q squared.
So will
That
it
equation.
be like this, like
They'd be exactly like
this,
that.
like how it was before we filled it in?
Exactly. Except I don't give you the equation.
Okay.
You're going to come up with a formula, but it's going to look very much like that. Okay? All right. Complete a Punnett square given the parents' blood types. Now remember, I have to give you the parents' genotypes, right? You're going to draw, well, the Punnett square is actually drawn on the test for you, so it's that square. So you're going to take the one gene from the one parent and the other gene from the other parent, and you're going to give the genotypes. So again, you're going to be able to tell me that, so you're going to complete the Punnett square, just like we did in class with those examples that I gave you. And you're going to be able to tell me what are their genotypes and what are their phenotypes, right? So genotype is what you're actually coming up with in the Punnett square because you know what the parents are. So you know that's the genetic makeup of the child. But if you took that child and tested them in the laboratory, what blood type would you get? In other words, if they're an AO, they are just an A. If they're a BO, they're a B. They're an OO, they're an O. Make sense? Okay, so genotypes and phenotypes. Match the immunoglobulin characteristic to the correct immunoglobulin type. So just that one page like in your PowerPoint where I have IgGs and IgMs, I'm going to give you the characteristic. You're going to tell me whether it's IgG, IgM, both, or none. Okay? What factors influence the antigen-antibody attachment? So it's that PowerPoint slide on what we talked about today. How many antigen-antibody attachments? So again, how you're forcing it together. You know, I can go back and look, but do you know which PowerPoint I'm talking about? You'll see it. Antigen-antibody attachment. How are antibodies made and what are they made of? So where do your antibodies come from? B cells. And what are they made of? So think of your SPE in chemistry.
Plasma cells?
Plasma cells make them. What are they made of? So think about your SPE in chemistry. SPE, first fraction was albumin. Second fraction was alpha 1. Third fraction was alpha 2. Then you had beta. What was your last fraction? Gamma globulins. Remember we did the SPE. We separated out our proteins. If the gamma globulins were increased, you then had to do an IFE, right? And that was all about your antibodies.
So it's
The gamma globulin fraction of your SPE, serum protein electrophoresis, is your antibodies. Remember? Way back when? Pulling it out of the cobwebs?
So it's made of gamma globulin.
You got it. Yep, gamma globulins.
Okay.
What is immunogenicity and what influences it? Immunogenicity is how much is that antigen going to cause a reaction? And what influences it? How many exposures you had? How much blood you got exposed to? Right? How is complemented, activated, and destroyed? How did we say we destroyed it? How did we destroy it?
We're just deactivating it, aren't we?
Deactivating it. EDTA, heat, right? Right? And how is it activated? Just simply in that complement cascade, just like the dominoes falling, causing it to react. Who regulates blood banks and what is needed to be safe? So, who were the ones that we said came in to inspect your blood bank?
Okay.
CAP, JACO. Which ones were significant for blood bank? FDA, Postal Service. The main one I want you to key in on was the FDA. Why does the FDA care what we do in our blood bank? It's like keeping your lunch meat cold enough in the deli, right? I mean, it is a food product, if you will, a medicine, if you will. I did ask my supervisor yesterday, and I said, has the FDA ever come and inspect the blood bank? And she says they have every right to. She says, but all they do is about every year or two send her a sheet and ask her, are your temperatures okay? Is your blood, do you manufacture this? Do you do this? And she says, it's just a yes, no sheet. She says she physically does not remember an FDA person ever walking into the lab. But they do send her a sheet. And then, of course, she signs it and says that she guarantees it. That's what that is, so that they could come in and ding her if, for some reason, she says that she doesn't ever recall having an actual FDA person. I was just curious. All right, no student expectations in blood bank and how to stay out of trouble. All right, so what do I expect for you? How much of your ABO do you have to get correct? Performing it, not test-wise, performing it. ABO is 100%, yes, all the way through the end, yes. And how do you stay out of trouble? Ask lots of questions, yes. Now, as far as your reagents go, I gave you a box of reagents, right? I asked you to put your name on it. Why was that significant to me? Why did I care? Why couldn't I just say, oh, go in there and grab whatever A or B? Because remember in the beginning, if ABO is that important, right? As when you do your quiz today, when you are done, you are simply going to leave all of your tubes in your rack. You're just going to turn your test paper over on top of your rack. You're going to leave your reagents on the desk. I'm going to come through and I'm going to look at your tubes and see if your reactions match what you should have gotten, plus what you wrote down, does it match what you saw? Okay? If for some reason you got a reaction that I was not expecting, I will repeat it with your reagents. So that way I know that if for some reason your A came up positive and it's like, oh, this should not be, I use your reagents and lo and behold, somebody put something else in there and I would throw out that. So if you're getting a reaction that I'm not expecting, but it is what you got from your box, then we can narrow it down. If you borrow somebody else's and I don't know about it and you get a reaction that doesn't match what I do, I use what's in your box and I get what I should have because you weren't thinking and you borrowed somebody else's, then I can't grade you right. Make sense?
So the attachment sheet that you're talking about,
I'm sorry?
the antibody and antigen attachment sheet that you was talking about, where you list the factors.
Uh-huh. Where's my PowerPoint? So factors that determine antibody production is page 22. Let's see. Where's the other one? I don't know why mine are all out of order. Well, I'll find it. But you guys can go ahead and go, because I know you got to go. So you guys go do whatever you want, and then I will find it for you. Sorry, my PowerPoints are just all out of order, and straight. But anyway, I need a few minutes to get your practical ready, so we'll start at 10 o'clock. So you can go eat, you can go to the bathroom, you can do whatever, and then we are going to repeat everything we did on Tuesday before your quiz, but I want to have your quiz ready so that we can go when you are ready. Then when you're done with your quiz, you're good to go.